Hello, everyone, and welcome to today's webinar, Unleashing the Power of Automation for High-Throughput Antibody Synthesis. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. Today's presentation is interactive, and we encourage you to participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Now, without further delay, I would like to introduce today's speaker, Dr. Rebecca Moschall, Staff Scientist, Research and Development, Life Sciences Solutions, Thermo Fisher Scientific. For a complete biography on our speaker, please click on the Presenter tab at the left of your screen. Dr. Moschall, welcome. It's wonderful to have you here today. You may now begin your presentation.
So thank you, Christy. I really appreciate you hosting us today. And hello to everyone. Thank you for joining today's webinar about unleashing the power of automation for high-throughput antibody synthesis. I would like to start today's webinar with some core facts about Thermo Fisher and our gene-to-protein service. And afterwards, we will explore the discovery and optimization of antibody therapeutics, emphasizing the benefits of high-throughput compatible antibody production to accelerate discovery timelines. And next, we will then demonstrate the performance of our HTP automated antibody production platform and will further share some insights of our development approach. And finally, we will conclude by highlighting several applications of the automated HTP antibody production platform. We at Thermo Fisher are, of course, a very large organization with over 120,000 colleagues and with a strong industry track record, having a collective expertise in a multitude of areas, as for example, in talent, delivering results to you, and we further invest in innovation. As an industry leader, we understand that it's you, our customers, who are at the forefront of innovation, pushing the boundaries of what's possible in science and medicine. That's why we are here to support you every step of the way with our expansive portfolio of brand, depth of capabilities, and our comprehensive and scalable services. With that, we support you during all phases of clinical trials, pharma services, and much more. Among this plethora of services, Thermo Fisher also focuses on accelerating discovery research. One example of this category is our gene-to-protein production service. Our gene-to-protein production service helps to accelerate protein R&D timelines and simplify processes to enable faster transition to commercial production. We provide here the complete service chain from gene sequence optimization and de novo synthesis down to protein expression, purification, and analytics. Using our workflow, you simply need to deliver your electronical sequence for your antibody of interest, and we will take care of the rest. So in detail, the first step of our gene-to-protein production workflow is the sequence optimization based on the chosen organism. The gene optimizer algorithm performs codon optimization without changing the underlying protein sequence and takes into consideration over 20 parameters to select the most favorable codons and avoid the occurrence of motifs that can reduce or hamper protein expression. Among them, as exemplified here, codon usage, RNA secondary structures, or also cryptic splice sites. The optimized sequence is then synthesized using our next generation gene synthesis technology and is then cloned into the desired expression vector and is further sequence verified by NGS. Correct monoclonal plasmids are then transfected further into mammalian cells, and the resulting recombinant antibodies are purified and analyzed according to your needs. Now, of course, one critical question remains. Why is it so essential for us to help accelerate antibody discovery and optimization processes? So the answer is shown here as a workflow. Customer needs to test earlier, and more antibodies are needed for screening to fulfill the needs of antibody development. Here in this figure is a simplified breakdown of the usual workflow for discovering and developing antibody therapeutics. After target identification, this typically starts off with the discovery efforts and the selection of a potential antibody against a target of interest. And this selection is later narrowed down to candidates with the most preferable characteristics. And those lead candidates are taken to the next phases for safety and toxicity screening, and later these selected antibodies enter clinical phases. So talking to our customers, we identified three critical needs here. They want to move faster from one stage to the next, and they want to minimize the risk for late-stage failure by identifying early the most promising lead candidates. And they want to generate high-quality data along this path to train their AI or machine learning-based models for better predictability. So to tackle the bottleneck of late-stage failure and the need to test earlier, one main goal is to accelerate the lead identification during optimization phase, which is here highlighted in purple when customers perform their screening campaigns.
Thank you, Dr. Moschall. This does bring us to our first audience poll. Now, our first question is, at what stage of antibody development are you most likely to outsource? Is it discovery, optimization, preclinical, all equally likely, or not applicable? Again, I'll give you a moment to choose your answer. Just click on the answer that fits best to the question of at what stage of antibody development are you most likely to outsource? Discovery, optimization, preclinical, all equally likely, or not applicable? Dr. Moschall, back over to you.
So for that, let's zoom into the lead identification. Traditionally, we have relied on two classical approaches for screening: in vivo immunization and in vitro preparation and testing for phage display libraries. However, a third approach is gaining prominence, the in silico de novo antibody generation. This method relies heavily on large data sets to train machine learning and artificial intelligence algorithms. And as a result, each of these approaches generates thousands of antibody sequences that needs to be synthesized. Unfortunately, many of these developability assays require only small amounts of antibodies. This presents an opportunity to run several assays in parallel and to miniaturize antibody production. And to address this need to accelerate antibody screening campaigns, we developed an automated platform for high-throughput production of ready-to-screen antibodies. In detail, what does this automated small-scale antibody production service offer? The first column shows that it guarantees a confidence in service. By applying design for Six Sigma principles, we developed an automated system with a minimal operator intervention and a high lot-to-lot consistency. The end-to-end traceability of the ordered product is guaranteed through full workflow integration in our manufacturing execution system, and seamlessly barcode tracking from DNA to final product is ensured. Further, our product enables customized antibody screening as displayed in the second column. Several possible options can be chosen: antibodies expressed in XP2 or XP203 cells, as well as the choice of supernatant product or a purified antibody product that further undergo buffer exchange and stereofiltration. Further, you may order up to 100 of antibodies at once. In addition to that, the data delivered from the antibody production platform gains a knowledge as an asset. To guarantee maximal confidence in data quality, all our QC instruments were validated via measurement system analysis based on ICH guideline principles. Our standard QC includes further the titer measurement in the supernatant, protein concentration, the calculated yield thereof, and the purity of the purified samples. Of course, also additional services are available upon request, like, for example, endotoxin testing. The automated antibody production service paves the way for a multitude of applications. So it allows a seamless transition to larger scales when more material is needed, and by doing so, the capacity and data quality compile with machine learning approaches. While at the same time, high product quality and quantity makes the product feasible for downstream developability assays, but also your internal resources are freed up to focus on your own discovery. But now let's take a step back to discuss how we ensure a streamlined, high-throughput antibody generation. We have developed a robust system with the following core attributes. First of all, the minimal operator intervention. This is achieved through our fully integrated manufacturing and tracking system. Further, the sample and data integrity is ensured via a seamless workflow and API exchange between the liquid handling and standalone devices, which is further mediated by our MES system. The traceability thereof is guaranteed by utilizing 2D barcoded tubes and barcoding gates, which are connected to the MES system, enabling that each individual sample in every workflow step is fully traceable. So the workflow itself begins with the synthesized DNA, which is stored in an automated fridge, which is shown on the left-handed side. When ready for transfection, the DNA samples are transferred to our liquid handling platform, shown here in the middle, for progressing the automated protein production workflow. After protein production, aliquots of samples are processed for quality control analytics, and the final product is stored within the automated fridge again. So taken together, this system enables that we can monitor the performance of each single subsystem within the integrated environment, and we can manage and integrate the data via the MES system. We can identify and resolve any potential bugs, and we utilize the subsystem outputs as in process control to monitor and to ensure the integrity of the process. So let's take a moment to revisit the earlier stages of the development phase. Before establishing the complete workflow, which was shown on the previous slide, we developed each individual piece or subsystem of our platform in accordance to Six Sigma principles. So why Six Sigma? Adopting Six Sigma methodologies ensure a lean design concept, allowing us to maintain maximum process control while achieving optimal performance. And this approach is crucial to deliver a robust and efficient automated system. So the left-handed side illustrates the Six Sigma principles that were applied during our development process. It all starts with a screening of significant factors. So each subsystem, such as transfection or purification, was screened to identify significant factors that influence the output. We employed here design of experiment methodology to ensure a comprehensive analysis. Once the significant factors were identified, we utilized predictive model to characterize how interaction among factors influences the response. And finally, the identified factors were then adjusted to their optimal set points. This crucial step allowed us to maximize responses, as highlighted in the middle figure indicated here by the arrowhead. And additionally, it ensured a robust operation, as demonstrated in the right-handed figure, where the readout remained stable across the entire range of factor A and B being tested, which is here indicated by the 4 arrowheads. So consequently, this results in a solid and reliable process being built. So in sum, by adhering this design for Six Sigma principles throughout the development phase, we ensure that our automated antibody production platform is not only efficient, but also highly reliable and capable of delivering consistent performance. But nevertheless, since flexibility is also essential to cater to diverse needs, we provide a range of options you can choose from. So one tailored option that our platform offers is the ability to select between 2 cell lines, XP2 and XP293. So XP293 are the most common cell line for transient transfection and provide robust yields for many protein formats, especially for antibodies. They are frequently used for antibody screening as well as immunogen production. Whereas XP2 cells, they are often used for stable expression and for proteins that are difficult to express in 293 cells. They are commonly used for the expression of antibodies as well as antibody derivatives. However, when choosing between these two cell lines, it might be important to consider individual harvest dates, as they result in a cell line-specific turnaround time. And this factor, of course, can influence the overall timeline of the project. Beside the cell line-specific turnaround time, depending on the downstream application, you may require either a supernatant product or a purified product. The supernatant product are often needed for expression screening and others, and they will result in a turnaround time for protein expression of 10 days for XP-CHO and 7 days in XP293, which is here highlighted by a light green and by light purple. Purified products, which are needed for many downstream applications, an additional day is here required to complete for purification and buffer exchange. So regarding our purified product, the upcoming slides will now offer you insights into our performance during the purification and buffer exchange development processes. When optimizing our automated purification workflow, we utilized integrated Kingfisher Presto devices in combination with PS Protein AG agarose beads. This methodology ensures nearly 100% binding efficiency to the beads, which is here highlighted by a blue box, and it achieves around 80% elution efficiency, which is indicated by the red box. And this in total culminates in an overall recovery rate of 78%, which is here highlighted by a green box. And here we used approximately 2 milligrams of protein. Next, our automated buffer exchange and stereofiltration processes are designed to minimize runtimes and dead volume loss, thereby also maximizing capacity throughout the automated workflow. The principle of the buffer exchange is illustrated on the right-handed side, showing that small molecules and salts pass through the resin beads, whereas the proteins, being too large to enter the resin, will pass around them. For that buffer exchange, only 3 steps are needed. First, the equilibration, then loading your sample and spin it down. This technique enables buffer exchange to be completed within minutes, significantly reducing waiting times compared to other methods such as dialysis. Moreover, the use of zebra size exclusion resin enables highly efficient buffer exchange, achieving nearly 100% depletion of our initial tris-glycine buffer, which is indicated by the blue and red boxes. And this ensures a high protein recovery regardless of the protein concentration being used, what is indicated here by the green boxes when we are using a protein concentration of 2.5 microgram per microliter, we achieve a recovery of 95%, whereas we are achieving 92% recovery when we are using higher protein concentrations with 1.5 microgram per microliter. So taken together, the combination of the automated transfection using the XP cell lines, the purification using the Kingfisher devices, as well as the buffer exchange using zebra plates results in a robust and highly reproducible method. So after integrating all subsystems into our final automated system, we next validated our platform using 38 antibodies which are well characterized, including chimeric, humanized, as well as human antibodies. In this testing setup, each antibody was expressed in triplicate and were randomly distributed across the expression plates. And further, we tested our automated system using both XP293 as well as XP-CHO cell lines, producing the antibodies in a 2.5 milliliter culture. But now let's dive a bit deeper into the data generated by the HCP antibody production platform. First of all, following the automated transfection of our antibody task set and the incubation thereof, we collected the supernatants and measured the antibody titers using biolayer interferometry prior to the purification. And subsequently, the purified products were then analyzed for protein concentration. So to validate the reliability of our workflow, we evaluated the data for precision within the same run and between different runs. For the XP293 cell line, shown on the left-handed side, Both the supernatant as well as the concentration data of the purified product exhibited a low coefficient of variation of approximately 4%, indicated here as a dark blue and a light blue box. And this indicates a high reproducibility within a single run. Additionally, the variation between two independent runs was also low, indicated here as a red box with a CV of 11.2%. So similarly, the XP show cell line, shown on the right-handed side, demonstrated also a high reproducibility with CVs of 9 and 9.5% within a single run and a CV of 9.8% between two independent runs. So in conclusion, our robust and reliable workflow ensures the delivery of consistent and dependable amounts of protein, of course, regardless of the order time slot. Next, this graphic here shows the correlation of the two independent runs shown on the previous slide, this time displayed as a XY plot performed in XP-CHO. The left plot refers to the antibody titer between two separated runs, and we are achieving a high correlation with an R-square value close to 0.9. After purification, buffer exchange, and stereofiltration, we of course introduce a bit more noise into the system, but still see a good correlation of higher than 0.8, which is shown on the right-handed side. So since we are measuring titer and concentration as well as volumes of each sample, we know that on average we recover around 70% of our sample with an average protein purity of 95%. But so far it was just a lot of data points. Next, we picked out two rather opposing examples of one humanized and one human antibody, which are here highlighted in blue and green. As expected, antibody expression, recovery, as well as purity can vary based on the physical or chemical properties of an antibody that might influence their interactions with protein A and their folding and dilution at low pH. For instance, antibody recovery was high in case of antibody A, but the purity was lower than average with 83%. Antibody B showed the opposing effect. Having a high expression and purity with 97%, they showed a lower than average protein recovery with around 48%, but it still manages to achieve the higher yield. When we examine now the data set for the alternative cell line XP293, we observe an even higher reproducibility between runs with an R-square value exceeding 0.9. Revisiting now our two antibody examples, we note that the characteristics in purity and recovery exhibit a high reproducibility, underscoring here the robustness and reliability of the production platform. However, the expression levels in the two different cell lines vary, with an overall yield of antibody A surpassing that of antibody B. And this is due to the more efficient expression of antibody A in XP293, while the expression levels of antibody B remained more or less consistent between the two cell lines. And these results taken together highlight the critical importance for protein and cell line specific insights which are needed for efficient antibody screening. Our high-throughput antibody production platform effectively provides this insight and ensures sufficient amounts and purities for conducting multiple developability assays from a single sample. But what should you do when you need more protein to address an increased number of downstream applications? So scaling up in this case is feasible. This case study here exemplifies the expression levels of three antibodies expressed in 2.5, 200, as well as 1,000 milliliter scales. The data shows that the relative levels of expression remain consistent when comparing three different antibodies, which are here indicated by different shapes and are expressed as triplicates. And they're expressed across small, mid, and large scales. Small scales are plotted here on the x-axis, where mid and large scale are plotted on the y-axis. In this case study highlights that the relative expression between antibodies stays constant across different scales. So this means high expressing antibodies remain the high expressing across all scales, indicated or exemplified here, for example, in XP293 by the squares. However, the absolute expression may vary across scales and between cell lines depending on your antibody of interest. For instance, in XP293, as displayed on the left, absolute expression level may vary across different scales, which is indicated here by the dashed line within small scale at 400 nanogram per microliter expression and mid and large scales being around 800 nanogram per microliter. In contrast, XP shown on the right-handed side, expression levels are comparable across scales for each of our selected antibody candidates, though this may vary depending on the protein being chosen. So this consistency makes it easy to reorder or upscale production if more protein is required for follow-up studies. As we now approach the end of today's presentation, I would like to take a few minutes to summarize the key aspects of our HTP antibody production workflow. Our gene-to-protein services allow you to send us the digital sequence of your antibody of interest, and we will handle the rest. By applying sequence optimizations to enable maximal expression combined with miniaturized HTP antibody production, we ensure sufficient amounts of antibodies for several downstream applications. But moreover, we offer flexible options to meet individual needs based on your antibody candidate. Whether you need to choose between our two cell line systems, adapt format options, or apply different add-on services such as endotoxin testing, we have you covered. Regardless of the flexible options you choose, we guarantee further consistent production of antibodies, ensuring high-quality data that can mitigate late-stage failures and reduce long-term development costs. And further, the proven consistent relative expression between antibodies across scales ensures seamless integration of our services and facilities scale up for later development efforts. With that, we have reached the end of today's webinar. Thank you for your attention and the interest in our product.
Thank you, Dr. Moschall. Now this brings us to our second audience poll. Would you like to speak with a technical sales specialist about options for gene synthesis and protein expression services? Please select yes or no. And again, that question for our audience is, would you like to speak with a technical sales specialist about options for gene synthesis and protein expression services? Select yes or no. Dr. Moschall, back over to you. Thank you, Dr. Moschall. And it does bring us to our third and final audience poll. Question for our audience is, are you interested in getting a quote on a protein expression project? Please select yes or no. And again, you'll see our question, are you interested in getting a quote on a protein expression project? Select yes or no. Now back over to you, Dr. Moschall, to close us out.
Thank you for your attention, and thank you, Christy, for giving us the opportunity to hold this webinar today. And I'm now more than happy to answer your questions.
And thank you, Dr. Moschall, for your informative presentation, and welcome back for our live Q&A portion of our webinar. Now, we do have some interesting questions from our participants that have already come in, but to our audience, if you haven't already done so, we invite you to submit your questions now. Simply type them into the Ask a Question box and click Submit, and we will answer as many of your questions as we have time for. So, Dr. Moschall, I'm just going to dive right into the questions here and start with this one. What is the maximum protein yield or concentration achievable using the high throughput protein, the HTP platform?
So the expression levels are, of course, depending on the specific antibody of interest and the chosen cell line, as we learned today. In our test with the antibody test set, we achieved antibody yields up to 1.5 milligram and final protein concentrations of up to 5 microgram per microliter. However, depending on your antibody, the maximum theoretical yields that the system can deliver is approximately 3 milligram.
Thank you. Now, our next question is, how can I determine the most suitable cell line for expressing my antibody of interest?
Yeah, thank you. This is a good question. So cell lines must be tested due to variations in the expression levels of individual antibodies, as we have seen today. For example, screening campaigns, the 293 cell line offers the advantages of authentic post-translational modifications and a shorter turnaround time, which enables to advance more rapidly through campaigns. But for poor expressors, switching to the CHO cells might be beneficial. It's also advisable here to verify the high expressors in CHO cells on a small scale before deciding to upscale production. So what we have seen throughout our development, we encountered candidates that expressed well in both cell lines, as well as those that exhibited high expressing characteristics in one cell line, but low expressing characteristics in the other, as we have seen with the antibody A and B candidate. So therefore, depending on the selection criteria, whether this is screening for the highest expressing antibody or selecting a candidate for scale-up production, the choice of the cell line, of course, may vary.
Thank you. Now, Dr. Moschall, this question has come in a few different variations. So what measures are taken to maintain a sterile environment during that transfection process?
Yeah, good point. So the DNA preparation and transfection are conducted in a sealed and sterile environment. So to prevent any microbial contamination here, the material is introduced and removed via kind of an automated lock module on the liquid handling platform. And of course, in addition, we employ regular automated UV cleaning and maintenance cycles. And with that, we ensure high quality standards. And in addition to that, we also demonstrated that our final products are free of endotoxin contamination.
Thank you. And we have time for one more question. How does DNA synthesis enable and support high throughput protein production?
So we have several ways to accelerate the antibody production via fast DNA synthesis for your antibody of interest, for example. So the efficiency of synthetic antibody production is increased here by using a common sequence for the constant part of the heavy or the light chain. And this allows for the coding sequence of the constant part to be integrated into a customized vector backbone, meaning that this part of the expression construct does not have to be synthesized de novo for each new antibody. But instead here, only the variable part of the sequence needs to be synthesized and is then cloned into this backbone. And when we take this together, this results, of course, in a more efficient process with a higher success rate and lower cost. And when we estimate our processes here, we can process up to thousands of samples in parallel, and our average turnaround time is 8 business days, for example.
Thank you again, Dr. Moschall, for this excellent Q&A session and for your time today. This does conclude our session. Now, before we go, I do also want to thank our audience for joining us today and for their interesting questions. Questions we were unable to answer today, and additionally, those submitted during the on-demand period, they will be addressed by our speaker via the email address you provided at the time of registration. I would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. I do want to bring your attention one last time to the survey on the left of your screen. If you missed answering any of the polls during the presentation, you can select your answers now. And additionally, if you are interested in getting a quote on a protein expression project for your future research, check that yes box. Until next time, everyone, thank you for joining us. Take care and bye-bye.